Good afternoon and selamat sejahtera, everyone. So, host, can we start now?
I think give another few minutes.
Okay, all right.
The participant only 63.
All right, okay. We'll wait and we'll wait for your cue. So, good afternoon and salam sejahtera to everybody who has joined us today for the International Scientific Webinar 2025. So, please allow me to introduce to you our speaker for today, TS Dr. Angeli Ambayya. Dr. Angeli Ambayya is currently a scientific officer with the Ministry of Health, Malaysia. She is also the founder and director of El Dunamis Scientific Academy, founded in 2025. She was a former visiting scientist at the Royal Melbourne Hospital, Australia, and research associate at Newcastle University, United Kingdom. Angeli specializes in flow cytometry, genetics, and genomics of hematology malignancies. She is trained in flow cytometry and stem cell transplantation at the Royal Melbourne Hospital in Australia and the University of York in United Kingdom. She obtained her certifications in genomics and precision oncology from Harvard Medical School, USA, and underwent bioinformatic training at Newcastle University, United Kingdom in 2019. Angeli led several MOH Malaysia research projects, including hematological reference intervals in a multi-ethnic population, which included 2,725 subjects nationwide. She was also the project leader and principal investigator for several genetics and genomic projects involving patients with acute leukemia. She has been serving on the Molecular Analysis Scientific Committee for the International Society for Laboratory Haematology since 2019 and holds the position of Vice Secretary for Persatuan Pegawai Sains Malaysia. Dr. Angeli is here to present us with a very interesting topic. NGS has become like a household name in the laboratory scene. So she's here now to introduce to us, or the topic of her presentation will be Introduction to Next Generation Sequencing Technology. So without much ado, Dr. Angeli, the floor is yours. Thank you.
Thank you very much, Dr. Sashi, for the kind introduction. Good afternoon, everyone. Please allow me to share my screen. Okay. Can everyone see my screen? Dr. Shashi, is it visible?
Yes, it is visible now.
Okay. Okay. Good afternoon, everyone. The topic of my talk today is Introduction to Next Generation Sequencing. Okay, so let's move on to the outline of my presentation for today. So these are the outlines. First, I will start with fundamentals of NGS and its application, and discuss why is it such a powerful tool in the field of genomics, and suggestion on how you can design your NGS experiments. And by the end of talk today, you will be able to understand how NGS works and some of the cutting-edge applications. So let's move on to the introduction. What is next-generation sequencing? NGS is also known as massively parallel sequencing, which is a high-throughput sequencing that enables rapid sequencing of different types of nucleic acids, including the DNA, RNA, epigenetically modified bases, by generating millions to billions of sequence reads simultaneously. Unlike the traditional sequencing, which reads one fragment at a time, NGS can process large numbers of molecules in parallel, making it scalable for small targeted studies up to whole genome and transcriptome sequencing. So there are several basic steps in running an NGS experiment that includes the sequencing of the nucleic acid, followed by the alignment of the data, assembly, and finally the data analysis and clinical interpretation if you're running in a clinical setting. So what are the applications of NGS? So it encompasses different applications for different nucleic acid, including DNA sequencing, where we can perform the whole genome sequencing, whole exome, meaning that it covers the coding regions of the genome, targeted panel, and variant discovery. For the RNA sequencing, we can do the gene expression profiling, transcript isoform discovery, fusion gene detection, non-coding RNA analysis. As for the epigenetic sequencing, we can do the DNA methylations by performing bisulfite sequencing, chromatin accessibility studies by using ATAC sequencing, histone modification, which is also known as the chip sequencing. Next, we can also use for the metagenomics and microbiome profiling by performing sequencing on mixed microbial communities without culture. And the latest trending sequencing technology is single-cell sequencing, where we can perform a single-cell resolution nucleic acid detection on the DNA, RNA, and epigenomes. So I've summarized the next generation sequencing application in a flowchart. So for the DNA sequencing, as I've mentioned just now, we can perform the whole genome, exome or targeted panel, which will lead to the identification of single nucleotide variants, insertion and deletions, also known as indels. And if we are using the longer read sequencing, we can also detect the structural variants. On the other hand, we can also perform transcriptome sequencing by either using the total RNA or only the mRNA, which can yield to information such as for gene expressions, fusion genes, differential transcript usage, RNA editing sites, and express variants. And it's something like a variant calling on the using the transcriptome sequencing data. Next, we can perform the epigenetic sequencing, single cell sequencing, and metagenomics. The focus of my discussion for today will be the DNA and also the transcriptome sequencing. So let's get some introduction to what are the sequences that are available in the market. So most of you are familiar with the short read sequencers such as from Illumina, MGI, and Iron Torrent. So the throughput ranges between low, medium, and high. that can generate between 25 to 100 gigabase of data. And the cost actually depends on your targets or variants that of interest, or if you're going to perform a whole genome or transcriptome, it should be a bit higher. And the read length is between 100 base pair to 250 base pair with an error rate of less than 1%. So these are the leading sequencers in the market currently. And there are even, the sequencing is revolutionizing, so there are more sequencers in place. So I'm just mentioning the main players in the market. So these are the reads that we obtained from the short read sequencing. So the read length, as I've mentioned just now, is about 100 base pair to 250 base pair. And we have the reference genome. So the tilings of the reads takes more effort because the reads are shorter. So let's look into the long read sequencer. The throughput ranges between low to high. And previously it was said to be more expensive than short reads, but recently I've come across adaptive sampling sequencing, where the cost was pretty much lower than the short read sequencing. So it depends on your experiment and the objective of your study. And the read length is much higher compared to the conventional short read sequencer. For the PacBio, it ranges between 15 to 20 kb, whereas for the Oxford Nanopore, it ranges between 10 to 30 kb. And with time, the accuracy has improved tremendously with the Q20 to Q30, which is the good quality read, with the sensitivity of more than 99.9%. So when you do a long read sequencing experiments, as you can observe from the diagram over here, the sequencing reads are much longer. So it is the outputs are longer contiguous sequencing, and it is easier to align to the reference genome because it is longer, and the alignment takes lesser efforts compared to the short read sequencing. So what kind of testing can be done using the NGS? So firstly, we can do some risk assessment and screening. For example, non-invasive prenatal screening by NGS, carrier testing for severe childhood recessive disease such as Mendelian disorders, and germline cancer risk testings. As for diagnosis, usually it is done in many types of cancer, but I'll focus on the hematological disorders such as leukemia, lymphoma, and thalassemia. NGS can also be utilized for prognostication, where we can discover biomarkers that could determine the patient's outcome. Next is for therapy selection, because there are targeted therapies developed based on the from the genetic profilings. So there are like mutations that are targetable by there are mutations that are targetable currently, especially in certain disorders such as acute myeloid leukemia, as I mentioned in the slide. So what are your sequencing strategies? In terms of comprehensiveness, whole genome has a better coverage compared to whole exome and gene panel. And the cost differs between it will be more expensive to perform a whole genome compared to a gene panel, depending on your experiment design as well. And now we can also perform a whole genome long read sequencing, which is able to detect structural variants. And as for the gene panel or whole exome or WGS using the short read sequencer, we are able to detect the single nucleotide variants and indels. So the TAT, obviously, the whole genome will take longer time to be analyzed compared to gene panel. So next, there is another concept that we need to familiarize today, which is the sequencing depth. Okay, read depth is crucial in making the tiling process a success. The higher the overlap, the higher the alignment confidence. For example, I've depicted a diagram where we can look at gene A, gene B, gene C, and other lowly expressed genes. So as we can see in the 5 million reads, the number of reads that covers each of the genes are lower compared to 50 million reads. So when this happens, the higher the read counts, the better coverage for the genome, and so we will be able to detect lowly expressed genes. So depth plays an important role in heterogeneous sample, especially in cancer studies. For example, you can see here the raw sequencing data. If it is just 5x, there is only limited coverage. At the same time, it is 10x, there are better coverage in the when we look at the comparison with the reference genome. So how do we determine our sequencing depth? So there are references that we have obtained from the Encyclopedia of DNA Elements, also known as ENCODE guidelines, which was published in 2003, which aims to identify all functional elements in the human genome and also other eukaryotes and bacteria. So where they suggested for eukaryotes, the million, if you are looking at transcriptional profile, like for example, like gene expression, the minimum sequencing depth that is required is for eukaryotes is about 30 million reads, whereas for bacteria, it would be about 10 million reads. So the experiment purpose is very important. If let's say you are planning to do a discoveries type of study, the minimum depth required is about 100 million to 200 meets for lowly expressed transcripts discovery. So next we can look into an aspect. Just now I was mentioning about read depth. Now we are looking into whether you want to do a single end or a paired end read. So what does it mean by single end or paired end read? So just imagine if this is a DNA molecule. It is about 300 base pair long. So if you are performing a single, if it is a single end sequencing, it is covering from one end of the DNA molecule. Whereas if you're using the paired end sequencing, it is covered at the both. So it is more specific and the detection is much better, especially for lowly expressed genes. And also it is able to reduce the false positive rate. So what are the platforms that have paired end reads? For example, it is Illumina and MGI. Whereas for Ion Torrent, they have a different principle altogether where they only perform single end sequencing. And so far we have been using Ion Torrent and the accuracy and the reliability is pretty much reliable to the other platforms. So if you are planning to run an NGS experiment, it is very important to familiarize yourself with Linux. Linux is an open source operating system, which was released in 1991. And the popular software that is operating system is called Ubuntu. So why do bioinformaticians use Linux? Because all the core bioinformatics tools were written in Linux. It supports many different programs, programming languages, and it handles multitasking very well. So if you're looking at a Linux PC, we don't call this as a folder, we call it as a directory. So we type in the commands using an interface, we call it as shell. So we interact through the terminal. So this is how we type in the commands and the programming languages over here. So this is example of basic Linux commands. So this is the first level directory, second level directory. And usually what you do when you open a PC, you will just double click the folders and you'll access to the different folders. But in Linux, you need to define the directories from the root directory to the first level, second level, and the levels of directories that you have in your system. For example, if you're using programming languages, so CD refers to change directory, mkdir refers to make a new folder. So these are the basic commands. Then if you are a biologist and you're interested to learn a bit of bioinformatics to ease your data analysis, it is better to pick up some programming languages. So where to get help if you are running an NGS experiment? My favorite is either BioStar or GitHub. So they provide, you have a lot of Q&A when you can post your questions, and there are experts there who can actually help you to answer questions on the biology part and also the bioinformatics part. So another tool that you need to familiarize yourself is the R Studio or R programming. So it is a S language. S language basically means it's a statistical language which was developed by AT&T Bell Labs. So basically it provides variety of statistical and graphical techniques and is highly extensible. Even for statistical analysis, some of us are running, instead of using SPSS, R programming language is becoming more and more dominant nowadays because it's very flexible. So where and how to use R? You can either download the R Studio if you're using a Windows PC, or if you have a Linux, you can run it using your terminal. So let's move on to the next concept, which is pipeline. So if you're running an NGS experiment, it is often they refer the workflow to a pipeline. So what does it mean by pipeline? So it basically means integration of different softwares and tools for your analysis. For example, if you are doing a raw data QC, the raw data is often referred to as a FASTQ file. I will discuss that more in my next slides. So the tools that we'll be using is a FastQC tool and also FastP Master, which is for quality profiling, adaptive training, read filtering, and base correction. Because mind you, these are sequencing data. So there are a lot of things that we need to trim out from the original sequencing reads. So we require different tools for the QC purposes. And next we will do the data alignment. There are two methods. Either it's alignment based by referring to a reference genome, or it's alignment free by not referring to a reference genome. So for example, if you want to do a reference based alignment, so we can use tools like STAR, HiSET2 and so on. If we are doing alignment free, then we can use Callisto and Sermon. So for example, for differentially expressed genes, we then need to do either a feature count or a TXT import to do the gene level quantification. And DEG, which is the differentially expressed gene analysis, we will require another pipeline known as DE-seq2. And finally, followed by downstream analysis using gene set enrichment analysis for detection of what are the pathways that are affected in the and perturbed in the expressed genes. So let's look at what does it mean by a FASTQ data. So for example, these are the reference genome. So there are fragments of short reads over here. So they are stored in a format that is known as FASTQ files, which is in a TXT with a format which contains tens of millions of long read lines. So for example, this red box over here refers to one read. So it is typically between 36 to 250 base pair long, and it consists the information of the sequence, identifier, and quality of each nucleotide over here. So how do we read a FASTQ file? So this is the raw sequencing data, the one that I've shown just now. This one single read. So I've enlarged it over here. So this is the starting symbol. This is the label for the sequence. And this is the starting position of the sequence. So we have a spacer, and each of these reads have a corresponding score according to the American Standard Code for Information Interchange. So basically a score of more than 20 is usually acceptable, and for clinical it is more than 30 because we are more stringent in accepting the results. So these are the codes, and usually we will use this data and we will upload it into the FastQC tools to filter out the reads that we need. So the next concept that you need to familiarize if you're running an NGS experiment is the Phred score. Phred score is basically measuring the quality of the identification of the nucleobases. So for example, if a Phred score of 10 indicates that the probability of incorrect base call is 1 out of 10, which is accuracy of 90%. If it is 20, then 1 in 100, which is 99%, which is usually acceptable in research and also in clinical, depending on the lab's policies. And Q30, which is 99.9%, which is the cutoff for most of the clinical labs. So the lower the Phred score, the chances of inaccuracy is higher. So as I mentioned just now, so we use the FastQC tool to remove all the low quality data. For example, you can see 3 color codings here. The green indicates it is acceptable. Yellow, just be cautious. Red is unacceptable. So usually when you're doing sequencing, towards the end of the sequencing reads, there'll be a lot of poor quality reads which we can remove using the FastQC package. So next, what we have to do is to remove the adapters. So usually when we have a DNA fragment, it will be attached together with the adapters, which will ligate the DNA fragments, and also we will have the index, which is the barcode. So barcode, because basically when you're doing sequencing, you're not just going to sequence one sample in a chip. In my setting, we usually sequence 16 samples together. So each of the samples will have a barcode. So the barcode is actually a sequence of the sequence that is given by the suppliers. So when we do the sequencing, we will identify each patient's or each samples based on the index. So what we need to do is to remove the adapters that attach the index before we can use the data for analysis, because we need to remove all the noises that comes from the adapters. So these are before the removal of the adapters, and these are after the data cleaning. So next, what we need to do is alignment. So alignment, so mind you, as I mentioned just now, there are a lot of fragments of reads that comes from the sequencer, depending whether it's a long read sequencer or a short read sequencers. These are fragments of genomes, fragments of read that we need to, it's like a jigsaw puzzle where you have to organize all the reads, and like you are doing a jigsaw puzzle assembly. So what we need to do now is to have to organize all the short reads and assembles them into a continuous chain as shown in the picture. So this is what we mean by data alignment. So there are several methods to perform data alignment. So for example, if you are using HiSET2 or star pipelines, so what we do is to align the reads to a reference genome. Whereas if you are doing another way of analyzing alignment is by doing assembly by de novo method, for example, Callisto and Selangor pipeline. So there are two ways, and it requires large amount of memory and CPU. And DNA and RNA sequencing and even methylation involves different types of software for analysis. So when we are doing the experiment, we need to be very clear of the objective, the type of sample, so that we can decide the right pipeline. So when we mention about reference genome, so most of you will be familiar with reference genome if you're doing NGS. So where does it come from? So we need to just an overview of reference genome. It is a haploid mosaic derived from 13 donors, later became 20 donors. The samples were obtained from Buffalo, New York, USA, and 70% of the reference genome came from the African-American descent. And the individuals who enrolled for the study had high risk for diabetes. And the remaining about 30% donors came from 10 samples and 7% from 50 samples from other sources. So the caveat emptor is the reference genome is not a healthy genome. The reference genome is not the common reference genome. And the reference genome is not the longest genome because it comes from several donors. And the reference genome is not a structured genome. And the reference genome is far from being perfect. So these are the caveats that you need to consider before performing the NGS experiment and when you are doing your variant calling and before you call something as normal or abnormal based on the reference genome. So currently there are two versions that are being still used for the assembly, which is the GRC-CH37, more in clinical settings, especially for targeted panels. It is a single representation of multiple genomes, and anything that doesn't match will not be aligned. So if there is a novel segment of fragments that you want to align to a certain population, for example, population X, if it is not available in the reference genome, it will not be aligned. So your novel SNPs will not be aligned to the reference genome. The more recent one is the GRCH38, where it stores the alternate segments representing wider populations. For example, if you have a chromosome 1, there's alteration in chromosome 1. It will give you the details on the chromosome 1 in general population, in population X, and also population Z, which is a better way of aligning your data. And in our own setting in Hospital Ampang, previously we have done some reference genome for our micro experiments where we have done the assembly and we have seen a lot of variation compared to the common variation that is seen in the controls. So for example, our CNV's copy number variations tend to be larger than what has been reported. So many of you would have been familiar with the My Genome project, which has been carried out in Malaysia. It is a very good initiative. So when it has been completed, we will have our own reference genome, which will translate better to our own patients in our own settings. So next, I will share my experience with the NGS technology. These are one of the studies that have used the NGS technology, which is entitled Elucidating Genomic Profiles of AML or Acute Myeloid Leukemia in Malaysia. The objective of this study is to uncover the underlying genetic profiles and their pathways contributing to the clinical outcome of patients in AML. And this is a collaboration study between Hospital Ampang, Haematology Department, University Sains Malaysia, Newcastle University, UK, and Qatar University. So in this study, I've recruited the AML patients. This is part of my PhD project, where I've recruited the AML patients and performed targeted sequencing on all the patients, and also the transcriptome sequencing, which is a high throughput deep sequencing of 200 million reads per sample because I wanted to look at the lowly expressed genes. All the sequencing was performed using the NOAA-seq 6000 from Illumina. And I've followed by bioinformatics analysis to detect the differentially expressed genes, fusion genes, variants, and finally, all these findings were compared with the clinical outcome of the patients. So these are some of our publications which has been published recently, especially the gene expression profiling and pathway analysis in acute myeloid leukemia normal karyotype, which was just published like a few weeks ago. You can, if you're interested in gene expression profiling, you can read through this paper. So this is a summary of what I've published in that paper. In this study, I've looked at the differentially expressed genes in acute myeloid leukemia by using a pipeline known as DE-seq2, where I've performed the subgroup analysis of acute myeloid leukemia patients with the healthy group, the samples at diagnosis, and also after treatment, which is known as RX, and different genotypes, because FLT3 and NPM1 is a prognostic marker for acute myeloid leukemia. So I've grouped the patients based on their genotypes. And the last group is based on their survival, which is overall survival of below and above 5 years. And followed by gene set enrichment analysis using a tool known as web-based gene set tool analysis toolkit, WebGelStart, followed by PCR validation, and finally functional enrichment analysis. So what do we mean by differentially expressed gene expression? So for differentially expressed gene, the main goal of the experiment is to look at the differentially expressed genes between condition 1 versus condition 2, or it can be more than 1 or 2 conditions, where we look at a lot of comparisons and consideration that we need to do. For example, what are the genes of interest and the gene length, the sequencing depth, what are the variation between two conditions in terms of if it is a disease, what are the differences? Are you looking at different diseases, or you're comparing between the disease and the healthy controls, and the variation between the controls? Because if you're looking at leukemia, for example, it is a heterogeneous disorder. So we need to reclassify the patients even further and stratify them so that the conditions between A and B doesn't overlap, so that you can look at the downregulation and upregulation of the genes of interest at different, maybe in different cells, tissues, disease states, development stages, or culture conditions. So these are what gene expression basically means. It's like a switch in your body. So in certain condition, it is turned off. In certain condition, it is turned on. So we are looking at gene expressions, and we will look at the pathways that are affected because of the differing gene expression in the body. So this was my findings using the differential gene expression profiling of subgroup analysis, where we can look at this. This is what we call as PCA plot. So when we look, this is basically looking at the condition using a 3-dimension plot, where we can see in acute myeloid leukemia, which is the red group versus the blue, there are significant difference in terms of expressions. So in terms of comparing these two groups, about 2,000 plus genes were upregulated compared to, and 2,000 plus genes were downregulated compared to the control groups. So similarly, there are subgroups based on the genotypes, the conditions during diagnosis and versus after obtaining complete remission 1, and their overall survival. So next, what I've done is to perform the gene set enrichment analysis for each subgroup, and the diagram over here reflects the AML patients versus the healthy controls, where we can see the blue chart represents the positively enriched pathways. So these are the pathway analysis where we have performed using the gene set enrichment analysis. These are the negatively enriched pathways. So the top positively enriched pathway is the cell cycle pathway. So in this, the red ones are the affected genes. There are total 56 genes that were affected in the cell cycle pathway, and 99 genes were affected, downregulated. The green ones are the downregulated one in the cytokine receptor interactions. So what we have found from this study is that there were consistent upregulation of proliferative pathways, including the cell cycle and DNA replications. And in contrast, immune-related pathways such as cytokine receptor interaction, MHC antigen presentation pathways are downregulated. So this supports the notion that this patient has been diagnosed with leukemia, and by looking at their upregulation of their proliferative pathways, so that is upregulated, and their immune system has been downregulated. So this supports the findings of our study, where it is commonly seen where upregulations of cell cycle pathways and all the DNA replication will be increased in cancer patients compared to their immune counterparts. So the next analysis that I've performed is to look at gene expression profiling of patients at diagnosis and after attainment of first remission. This is to discover biomarkers for minimal residual disease monitoring. So we're looking at markers that are suitable for disease monitoring after remissions. So as we can see, the DX1 is the patient at diagnosis, which is green. It reflects the upregulation versus the green, which is the downregulations. How to interpret this? Basically, when they are diagnosed, these are the, for example, MYCN gene is upregulated, and after they have attained their complete remission 1, these genes are downregulated, indicating probably that this gene could be a good marker for monitoring the MRD of the patient. Next, I performed the variant analysis using the DNA. So the pipeline of my interest was MUTEC2, where I performed the variant calling, comparing them with a panel of normal patients versus the AML patients. And I performed the variant effect prediction, followed by Sanger sequencing, and each of the variants were classified based on the established guidelines from the American College of Medical Genetics and also American College of Molecular Pathologists. And I've also performed the orthogonal validations. Since it's a bit complex, I'll just, the methods simplified. So what I have done is to compare the patients, these the ML patients versus a normal. So if there is any mutations, what I've done is to examine the mutations against the panel of normal, and I will do the, perform the variant effect prediction and classify the variants based on their, whether it's a pathogenic, benign, or variant of unknown significance. So this is the simplified methodology of my study. So let's go back to the central dogma and genetic variations. So in order to classify the variants, we need to understand the central dogma of biology and how DNA is transcribed to RNA and translated into protein so that we can know the impact of each variant's pattern. For example, this is the strand of DNA. So if the variants happen to be in the enhancer or promoter 5' or 3' of the DNA strand, it could affect the protein levels or the protein function. Whereas if it is occurring in the exon regions, so let's say it is affecting the second codon, the nucleotide T. So if it is going to change to a C, it is encoding the same amino acid, so it is silent. Let's say if there is a change in the amino acid from cysteine to tryptophan, so it is a missense protein, missense amino acid. And let's say if it is translated from TCT to TGA, which is a stop codon, it will produce a nonsense protein, which is a premature stop codon. So the protein is incomplete. And let's say if it is a frame shift, meaning that there's an insertion of another T, insertion of another T over here, yielding to a premature stop codon somewhere along the line. So these are the things, terms that we need to understand in order to effectively interpret the effects of the variants for the protein production, and also to interpret whether these the certain variants in the genes are clinically significant or not. So the variants were classified based on the ACMG, which is the American College of Medical Genetics and Genomics. The variants are usually classified into either a pathogenic or likely pathogenic based on their evidence that favors pathogenicity and capable of causing diseases. Variant of unknown significance is limited or conflicting evidence regarding their pathogenicity. Benign or likely benign, where there is strong evidence against the pathogenicity. So if you're interested in knowing more, you can read this guideline published by the Su and Richards et al. Next, we have another guideline. This is for interpretation and reporting sequence variants in cancer, especially if you're working on the sequence variants in cancer. AMP ESCO CAP classification is a better one, where it classifies the variants based on the tier. So the tier 1A to 2D indicates variants of potential clinical significance, especially if it is tier 1A, 1B, that are FDA or EMA approved therapy or biomarkers that are included with consensus from experts in the field. So if it is tier 3, it is unknown clinical significance. If it is tier 4, it is likely benign or benign variant. So to know more, you can go through this guideline as well. So based on the discussion just now, I've performed the variant classification of my AML cohort. So I've detected a total number of 7,361 variants, and I performed the classifications based on the ACMG and also the AMP ESCO CAP tier system. And I've classified the variants based on different prediction tools to know whether they are novel or reported. There are many tools in the market that you can explore that I will discuss later in my presentation. So in summary, these are the variants that has been detected in my cohort. So these usually it will be depicted in oncoplot, where we can look at the names of the genes that are involved, the types of the mutations that are detected, in the patient on their survival. So these are the early death patients. The red ones are the overall survival of below 5 years, and the green ones are the patients with overall survival of more than 5 years. And these are the variant types. So DNMT3A in this case had the highest numbers of variants in my AML cohort. So I've also identified clinically relevant variants in Malaysian AML cohort. So these are some of the variants that have therapies that has been either on clinical trial or have been approved by FDA. So for example, IDH1 and IDH2 mutations that have FDA approved therapies. So all these mutations has been depicted in a heat map over here. So the cases which has this kind of targeted therapy, we can enroll them for clinical trials and so on. So next we move on to downstream analysis. Now you have the DNA or the RNA or the methylation data. What are the things that you can do with your data downstream? So first thing first, it is always important to compare whether what you have found in your DNA or any sequencing to be true positive. So one way of doing or looking at that is by using IGV, which is the Integrated Genomic Viewer. So you can view your BAM file, which is the line data file, using the BAM viewer, where you can look at the breakpoints such as SNPs and indels and deletions or so on, or even fusion genes using the in silico validation method. And next, you can decide whether you want to do a gene set enrichment analysis or overrepresentation analysis, where you look at certain pathways of interest and you want to analyze whether the genes are upregulated or downregulated in the pathway, and you can analyze further and how this expressions or downregulation or upregulation affects the pathway of your interest. You can also perform network analysis using tools like String or Cytoscape. And you can also perform gene ontology, which looks at the molecular function, biological process, and cellular component of the genes or analysis of your interest. So gene ontology or gene enrichment, GO analysis, basically is to annotate a particular gene or gene products in three aspects, which is the molecular function, looking at the molecular activities, cellular components where the gene products are still active, where you will look at the cellular components, or biological process. So you look at the pathways and larger process which affect the gene activities and how do they contribute to each other. So next, you can also do the pathway analysis. There are various tools that are available for pathway analysis. So you look at the series of chain chemical reaction metabolic activities. So these are the names of the pathways that tools that are available online for free. For example, Kyoto Encyclopedia of Genes and Genomes, KEGG, which is the most common one. Wiki Pathway, Reactome, David's tool, Panther, ShinyGo, WebGelStart. My favorite is WebGelStart because it is simple to use, which I'll show in a while. So the next thing you can do is to, if you're looking at a larger picture, network analysis is suitable, where you integrate all the known and predicted association between the proteins, encompassing both the physical and the functional association. For example, here we are looking at the apoptosis pathway. So these three genes over here, this is the genes that are involved in the regulation of apoptosis. The ones over here, the red ones, it is the intrinsic pathways, intrinsic apoptotic cascade involved in the intrinsic apoptotic cascade, and the green ones are the extrinsic apoptotic signaling. So this is it's a larger picture. So I will do a comparison between the pathway and also the gene networks. So if you're looking at the biochemical pathways, the databases are usually curated, and it gives an overview of biochemical activities and of the biological process, and human interpretation is required. So it shows the series of chain and chemical reactions, and the connection represent describable and quantifiable relationship between the molecules, proteins, lipids, and so on. And the enzymatic process is elucidated, and changes via perturbation are predicted downstream. So these are the examples of pathways, tools that are available for pathway analysis that I've mentioned just now. On the other hand, if you're performing gene networks, it represents the relationship, physical, regulatory, genetic, or functional interaction. It is useful in discovering relationship in a large data set. Example, if you have a large number of samples, it gives a larger picture. And it shows the cluster of genes or gene products with evidence of co-expression. The connection usually represents the degree of co-expression, and in-depth knowledge of the process is not necessary. So it just gives an overview, and the common tools are Cytoscape and String. So what does pathway analysis basically mean? So just imagine this is an air transportation network. So there are different airports in this part of the world. And let's say there is suddenly cut off of communication because of terrorist attack, something like an immune evasion. So let's say there is a terrorist attack in the main hubs of the airports. So all the connecting airports will be affected as well. So this is what we do in all these networks and also in the gene set enrichment analysis, when we look at different pathways and we look at the hubs that are affected. So these are the tools that are usually used for my gene expression studies or even for DNAs and so on. So we can look at the ORS presentation analysis, gene set enrichment analysis, and network topology based analysis. So we can even select the organism of your interest, and it has different functional database of interest. So you can use this tool, you can just explore with your own data set to know how does this tool work. So apart from performing gene set enrichment analysis, pathway analysis, and so on, we can even do Swiss model for homology modeling. For example, let's say you have found a variant or that you suspect pathogenic in this region. You can just do get the sequence of interest, and then you can do a Swiss model, which by taking the sequence of interest, and then you can dump it to the Swiss model program. And then it is reliable when you want to understand the protein function, especially when the structure, the protein of interest, the structure is not readily available. So you can do this, something like we call as molecular docking and so on, using this tool. So the take-home messages for designing an NGS project, example for a malignant hematology or myeloid project or any other project, first you have to know the biology of the disorder. Which level do you want to study? Is it the DNA, RNA, protein, or epigenetics? Then you need to do a thorough research, literature search on what has been done and what can be done. Design your experiment, and you have to consider the budget, manpower, technical and clinical expertise, because we work in hand with the bioinformaticians and all the teams. So we need to know how you want to design the experiments. And if you're a biologist and you want to do an NGS projects, first we need to learn the fundamentals of bioinformatics so that you can control the flow of your study. And you have to ensure that you pick up the right pipeline. If you're not running a clinical lab and you are planning to do an NGS experiment, you basically need to read up and know which are the right pipelines for the study, and best to stick to experts' recommendation from BioStar, GitHub, or Broad Institute. And you need to have a team of, if you're performing a clinical study, you need to have a clinician, laboratory personnel, and bioinformatician who can work together. And be open to redesigning your pipeline, because some of my pipelines didn't work in the first run. So I have to redesign, realign my objective, and be flexible. So a good NGS experiment requires good quality of data, solid pipeline, and right interpretation. So these are my references. With that, I would like to thank Persatuan Pegawai Sains Malaysia for giving me the opportunity to present my NGS work today. Thank you. Back to you, Dr. Sashi.
Thank you very much, Dr. Angeli, for that very insightful sharing. I'm sure all of you have gained a lot of information and insights from her talk. I would like to now open the floor for questions to Dr. Angeli. If you have any questions, please put them on the chat boxes and we will read it out to her. And to start the ball rolling, maybe I would like to ask you a question first, while others are, preparing their questions. Yeah. So, so you explained to us a lot about the different tools that you used for RNA-seq or DNA sequencing, for the matter. So I'd like to know, what is your preferred tool for doing variant effect predictions?
Okay, for variant effect predictions, generally we use the, either we can use the SNP-EFF, which is the variant effect prediction tool, or you can use the variant effect prediction tool, web. You can just Google web from Ensemble. So we can, if you have the VCF file, which is the variant, the file that contains the variants, we can upload it. Either we can use some scripting, or just get the VCF file, we can upload it to the system itself, the website itself, where you will perform the variant calling. So if you want to do the variant annotations, there is another tool that I prefer, which is known as Franklin by Genox, supported by Kaizen. So it does a very good prediction of the variants, where it gives the pathogenicity information. So these are my preferred tool, especially for clinical settings.
Thank you very much for sharing, Dr. Angeli. Is there any questions from the floor? This is a good opportunity to learn from the experts. So, don't hesitate, don't be shy, please ask questions. I've got 3 questions, so if no one's asking, I'm going to carry on asking. So maybe I would like to also ask you, what are the downstream analysis that can be done for differentially expressed genes? You have already explained some, but I'm sure that there are
Yes.
others that, you would like to share as well.
Okay, for example, what I've mentioned just now, for my case, I prefer to do a gene set enrichment analysis, where I look at the pathways that are significantly enriched. For some of them, there are another method where they look at the genes and then they look where the genes are. For example, they just pick up certain genes and then they focus on genes, whether it's overexpressed or underexpressed. But the right way of looking at a gene, when you're looking at gene expression data, is look at the overall picture of the gene by doing a gene set enrichment analysis. So you look at the overall set, the entire set of genes that are either upregulated and downregulated, and then you look at different pathways. So I use the WebGenStar tool that I mentioned just now, where you can just load in the genes of interest by giving the information whether it is upregulated or downregulated. And then when you just click the next step, it will pop up giving the enrichment score, whether it is positively or negatively enriched in which pathways. So it is very helpful.
Thank you, Dr. Angeli.
Sorry?
I heard somebody, maybe there's a question. Okay, still no questions. So I carry on with my third question. So this is just, I'm seeking your opinion really. So in terms of like bioinformatic tools available for, when we talk about diagnostic tests, right? And using, bioinformatic tools to analyze them, for instance. So an analysis of omics, WGS, WES, transcriptomics, or even proteomics, or using a in-house bioinformatic pipeline is going to take a lot of effort in coming up with that, and also expertise of multi expert groups, right? So especially an involvement of bioinformaticians as well. So, is that more user friendly to use? Is it more user friendly to use like AI based tools and softwares to do these analyses, especially when we think about the diagnostic settings? And also maybe you can explain in terms of pros and cons, because we know that these softwares are a little bit more stringent than,
So for example, right, usually when we are looking at clinical labs, when let's say in my own setting, so when we have the, like for example, we have an iron torrent and we have an Oncomine MyDot panel. So it comes together with the software. So the tools has been integrated and we just need to use the tools. For example, they have the Oncomine Iron Reporter and then they have the iron Oncomine Reporter or something like that. So it basically, we don't do any manual manipulation of the data, but it is limited because they are looking at the GRCH 37, because for clinical settings, they are looking at the previous version of genome. But what we can do, if you are really keen and you have the expertise, we can for example, if we're looking at certain disease of interest, we can, for example, if it is myeloid, there are pipelines that has been developed by certain publications. They have mentioned, like for example, they incorporated different analyses into Hamlet, that is a pipeline. So rather than we create a pipeline, right, we can adopt to other pipelines that has been established by other researchers. And then we try it on and then fine tune the script to fit our analysis. Rather than, because we need to have some guidelines to start somewhere. As I mentioned, we cannot simply just, okay, I like this pipeline, I want to use this, is not recommended. So we always need to go for the disease of interest. What are the other researchers are doing? Are they doing, for example, if I selected MUTAC2, I have a reason because many of the other researchers were using the pipeline. So if anything goes wrong, I can always go back to the forum and then ask for guidelines. So it is not recommended when we have no idea on what we are doing to start a pipeline, and then it is quite risky because if anything goes wrong, we don't really know where it goes wrong and how to rectify them.
Yeah. That's very insightful. Thank you very much. That's a very detailed answer. And there is one question now I see online now, from two now. So it's from Miss Asma. Let's go with that first. So hi, doctor. Regarding the GSEA, may I know what type of file that we need to upload to the WebGesalt website?
Okay, basically we call it as RNK file. So we can create the file on our own, because when you have the, basically it's just you need the gene name, the expression detail, we call it as a log2 fold change, and also the p-value. So when you have these three details, we can just create, you can even use Excel, and then you save it as RNK file, and then upload the file into the WebGenStar tool. So it will detect the file. Or you can even just copy the whatever that you have typed in your Excel. You can copy paste into Web Delta. If it is the list is not too long, it will still run the gene set enrichment analysis. It is very straightforward and it's very user friendly. Yeah.
Well, thank you, Dr. Angeli. I hope Ms. Asma that answered your question. So there's another question from Hanif Riza. So in your opinion, TS Dr. Angeli, how prepared are the MLT scientific officers in performing NGS analysis in Malaysia? This is putting you on the spot, but yes,
Oh
that's
my
the
God.
question. Yeah.
So how prepared we are, I think it basically goes back to the individual. Whether do you want to take up the challenge? So for myself, I was a flow cytometrist. So for me to do a NGS experiment, I have to go for different courses. During the pandemic, I was like spending time learning bioinformatics, coding, and so on. So it takes in a lot of efforts on our side because if you're a biologist, programming is not that easy because we are never taught to program anything. So I had to start from scratch. But along the way, it became quite interesting because you can use your expertise in different ways. And now we have like I mentioned just now for clinical, right? You don't really need to run bioinformatics, programming and so on. You can just look at the variants. And if you know your facts and your stuffs, you can start interpreting like whether it's pathogenic or it's not pathogenic or so on. So I think some of the officers are prepared, MLTs too. I have good MLTs in my lab, so they are prepared. So Mr. Hanif, I hope I have answered your question.
Yeah.
Based on the individual.
Yeah. Yeah, very stringent. I mean, very broad training in bioinformatics is needed in terms of like developing pipelines and things like that. Yeah, but yeah, I would say like in terms of NGS analysis, at least our scientists are coming up to the mark definitely or already there.
Yes, agreed. Yeah, because I think certain institutes they have started their NGS and they're doing quite well.
Yes.
So yeah.
In fact, even in my institution, Hospital Nukazisa, we are doing NGS and our scientists are all trained in doing NGS.
Yes, that's great. Yeah.
Thank you very much, Ms. Asma and Mr. Hanif for those questions. We still have two more minutes. If there is any questions, we will wait. This is your last chance to type it out. But otherwise, TS Dr. Angeli, it's been a pleasure, hosting you today, and it's been a very informative talk, and I think we all gained a lot from your sharing.
Most welcome.
Thank you very much, and do not forget to, if you have any more questions, she has shared her contact details. Please get in touch to get more clarifications, and also do not forget to fill up the your attendance in the link that has been provided by the host to get your certificate of attendance. With that, I thank you again, Dr. Angeli, and everyone for attending today, and hope that you all have a lovely evening, and we will see you again in our next webinar series, International Scientific Webinar Series. Thank you.
Thank you, everyone. Bye.
Bye.